Hello and welcome to the virtual poster presentation of the ISMB 2020. My name is Schubach and I'm from the Berlin Institute of Health in Germany. My poster has the title Predicting the Effects of Regulatory Variants by Deep Learning. Until now, most disease-causing variants are reported in and around coding genes, and only a small number were identified in regulatory regions. However, today the diagnostic rate is still below 50% for genetic disease. So to increase this rate, we need to understand more about the regulatory part of the genome. Unfortunately, we have limited tools for assessing the effects of regulatory variants. With massive parallel reporter assays, we are now able to measure tissue-specific variant effects. But evaluation of computational predictors show that also myriad annotations and scores exist non-consistently predicts experimental regulatory variant effects. So here we developed a deep neural network trained on active and non-active regulatory regions from DNA data for multiple cell lines. The training process is sketched here on the slide. By feeding our new trained model with new sequences, we can predict how active the sequence is in the used cell line. If we change the base of a variant in that sequence, our model will predict the new activity, and the difference between reference and alternative results is then the variant effect. This is also sketched out at the poster here. In the results section, we first verified if variants with high effects are enriched in open chromatin. We took all open chromatin region and closed chromatin with the same GZ distribution for all from a holdout chromosome that was not used for training, and measured all variant effects through an in silico saturation mutagenesis. Figure 1, shown here, shows that high effect variants are significantly enriched in open chromatin, so our model is able to detect important regulatory regions in the genome. A trained learner, especially a deep neural network, can be a black box, but there are strategies to extract the learning content. With integrated gradients, a tool called TF-Modisco, we are able to find important transcription factor motifs learned by our model. This is described on the lower part of the left side here in the poster down there. So this strategy makes now our model much more concrete. Finally, we try to predict experimental variant effects from 7 regulatory elements, measured by saturation mutagenesis MPRAs. Our model outperforms regulatory scores, as you can see in Table 1 here. Now I will show you the variant effects of predictions for the SORT1 enhancer as an example, and we go in there in more detail. So now I'm going to describe this complex figure here, figure 2. On the upper panel in figure 2, you can see the experimental derived variant effects by an MPRA of the SORT1 enhancer. Every bar is a single variant, and if the bar goes up, the variant activates, and if it goes down, it represses the transcript. Blocks of activity are usually transcription factor binding sites. In the middle panel, you see variant effect prediction from our model. When you compare it with the experimental effects, you see that the pattern looks really similar. So comparing this with this. Sometimes our model cannot predict the right effect direction, but recovers the importance of the sequence. For example, we could detect nicely an HNF4 alpha binding site marked in light red on the left side. We could also verify that the motif was learned by our model using the integrated gradient method. Other scores, shown then in the lower panel here, rely too much on conservation on this enhancer and correlate poorly with the experimental data. So I hope I could convince you that using short sequences with open chromatin and a relatively simple deep neural network, we are able to predict regulatory variant effects. Methods for DNA interpretation allow us to extract the regulatory mechanism from our model, identify also motifs from various known key players in cell type-specific transcriptional regulation. This is really a critical step forward in the interpretation of variants in the common and rare disease. Thank you for watching my presentation here at the ISCB 2020. If you have further questions, you can contact me by email written in the poster here. Thank you.